Okay. So
Namaste, madam.
so can you see the first slide now?
Yes, ma'am.
Okay. So I'm going to continue with the topic which we were discussing last time, where essentially, the basic the topic is energy recovery as well as nutrient recovery. And under energy, we looked at how methane can be produced from the wastes. That is, the wastewater sludge which is generated still contains a lot of carbon. And if there is a possibility to digest it and to recover energy in the form of methane. And the second part was recovery of nutrients such as nitrogen and phosphorus. So nitrogen is what we got started with last time. So I have only taken one slide from the last week's lecture. That is when you talk about nitrogen, it is both nitrogen and phosphorus. They need to be recycled. They need to be recovered from the wastes, which are actually the domestic waste waters, which are released because there is enough nitrogen and phosphorus there, and being important nutrients, you do not want to lose them to the environment, but rather you would like to develop technologies whereby they can be recovered from the wastewater. And some of the problems we identified were with nitrogen release, particularly ammonium. Ammonium is released into the waste, into our domestic sewage as for example, in the form of urea and also in the form of protein food matter which is released. So it causes eutrophication, that is excess of nitrate or excess of ammonium. If it goes into the wastewater, it can lead to eutrophication. That is algal blooms and other things are associated with that. And second, nitrates themselves are also poisonous. Some of these effects were explained last time. So therefore, a pre or a post-treatment process, like the second point says here, that a pre or a post-treatment process is therefore required in order to remove nitrogen and phosphorus from the effluent. Okay, so there are things which, you first of all, you have to know the microbiology a little bit. That is, what groups of microorganisms are capable of removing this nitrogen, collecting it in which form, and then only you can hope to apply what is their biochemistry, what kind of reactions do they carry out. And number three, whether it is possible to develop a method whereby such kind of microbiologies can also be accommodated in the existing wastewater treatment plant. So that should be your theme. So when you talk about recovery of nitrogen, the processes which we will be talking about will be first nitrification, where conversion of ammonium, which comes in the domestic sewage, can be converted into nitrate. Okay, so these terms, I think those of you who have taken Shri Krishn's wastewater treatment would be familiar with terms like nitrification and denitrification. So please understand them very, very carefully, because they will be used all through the various processes which I described. So what is nitrification? It is conversion of ammonium, which is in the effluent, into nitrate. This is an oxidative process. The second part is called as denitrification. And denitrification means conversion of nitrate into nitrogen gas. Okay, so when you talk about, for example, nitrogen recovery, your objective is to combine both nitrification as well as denitrification, because the influent contains ammonium, which needs to be converted to nitrate. In the second phase, the nitrate needs to be converted into molecular nitrogen. However, this reaction, as you will see in the other slides, that nitrate cannot be directly converted into molecular nitrogen, but rather nitrate gets converted first into nitrite, then to nitric oxide, then to nitrous oxide, N2O, and finally into molecular nitrogen. Okay, so it's not a one-step conversion, and that also means if there are 4 different reactions which are occurring in between, that means there are 4 different classes of microorganisms, or it could be only one microorganism which could carry out 2 parts of the reaction, and the other two could be carried out by a different group of microorganisms. So it will always be a consortium which will be responsible for converting nitrate into molecular nitrogen. And that is how, when you pump nitrogen back into the atmosphere, you say that you have recovered nitrogen now, because it has been released into the atmosphere as a nitrogen gas. So is this clear to everybody?
Yes, ma'am.
Okay, now there is another term which I have used here, which is called as heterotrophic nitrification. Now the meaning of heterotrophic nitrification is that, the term heterotrophy is different from autotrophy. Autotrophic are photosynthetic organisms which are able to make their own food, but we call this as heterotrophic nitrification process. Okay, that means there is an external carbon source which is going to be required during this process. Okay, that is the reason why we use the term heterotrophic. So external carbon source is going to be required to be put in so that now this process can occur. Okay, so just be mindful of why we are using the term like heterotrophic nitrification. Okay, and it's explained heterotrophic because an organic carbon source has to be supplied to provide the reducing power. Okay, because eventually, you do require a carbon also in the process. So in other words, for nitrogen recovery, the first thing which many people can try out is nitrification or denitrification. The second process through which nitrogen recovery can be carried out is called as NMOX process. That means anaerobic ammonia oxidation. How is it different from the first processes? Because nitrification process, that is conversion of ammonium to nitrate, is an oxidative process. It has to be carried out in the presence of oxygen. Okay, but when you say anaerobic ammonia oxidation, as the name suggests, that it has to be carried out in the absence of any, in the absence of molecular oxygen. Okay, and due to space constraints and economics, because you will see that the economics of this particular process is much better, and although there are some disadvantages which are associated with conversion of ammonia under anaerobic conditions, but still the economics are very good. Low sludge formation occurs. It can also handle very high ammonium load. Okay, so many Western countries, particularly in Denmark, which is practically a leader in developing technologies for the wastewater treatment, they have already implemented these NMOX reactors. Okay, so how is NMOX different from the first process is that you do carry out oxidation of ammonium. Okay, so ammonium oxidation, but in the absence of oxygen or under anaerobic condition. And in the first one, it is an oxidative process. You carry it out in the presence of molecular oxygen. The third way that it can be carried out, which is still in the trial stages, is called as demo process, that is direct anaerobic methane oxidation, because we will see here that conversion of ammonium to nitrate or recovery of nitrogen is coupled to methane oxidation. Okay, so I hope that all of you will also know how the processes are different. And now in the next few slides, we will see as to, what kind of microbiology is involved and what kind of reactions are actually going to be involved, and is it feasible or not? Has it been practically implemented anywhere? So all of these things we are going to see now. So is this clear to everybody until now?
Yes, ma'am. Can you please explain the last point again, the demo
Okay.
process? What
Okay,
it is?
demo stands for direct anaerobic methane oxidation. Okay, so essentially what you want to do is that you do want to, because it's a part of the nitrogen recovery process. So ammonium will be converted into nitrate. Okay, that is called as the nitrification process. Nitrification will be converted now to nitrogen. Now this particular part is a reductive path. Okay, and the first part, that is converting ammonium into nitrate, is an oxidative process. All right, however, because in this entire process you need to have electron acceptors. Okay, somewhere where electrons are going to be accepted from methane, and finally you are going to release the molecular nitrogen. That electron acceptor becomes methane. Okay, so you're going to convert methane now into, methane is going to be converted into carbon dioxide. So the entire process of nitrification and denitrification is coupled with methane oxidation. And this occurs under anaerobic condition. Therefore, the process is called as the demo process. So is this clear to everybody?
Yes, ma'am.
Okay, so just concentrate
Yes, ma'am.
on otherwise there are many the fundamental things which you're likely to miss out if you just lose your attention. So now the first thing which I'm going to talk about is the first process, which is called as the nitrification and the denitrification process. Okay, and again I will repeat, nitrification is whereby, which is shown in this yellow box, nitrification is a process whereby ammonium is converted into nitrate. All right, this process is an oxidative process, and ammonium to nitrate again is not a direct one-step conversion, but rather there are several enzymes which are going to participate in the process. And the first step is like conversion of ammonium into hydroxylamine, and then converting it into nitric oxide, into nitrite, and finally into nitrate. So all of these are oxidation reactions. Okay, what kind of enzymes are involved? First of all, ammonium. Now, I'll tell you what is the significance of these enzymes after discussing what enzymes are involved. So if you look at, for example, this, we say ammonium, or it could be taken as ammonia. So the first stage is participation of ammonium monooxygenase. So a single atom, as the name suggests, it's a monooxygenase. A single atom of oxygen is going to be added. Okay, so once you do that, you see oxygen appears here. That is the reason why these enzymes are called as ammonium monooxygenase. They add this. Okay, and then you have, for example, this is hydroxylamine. So you have hydroxylamine oxidoreductase. Okay, or abbreviated as OR, which converts it into nitric oxide. And then there are like nitrite oxidoreductases, or you have nitrite oxidoreductases, which finally converts it into this. Okay, so some of the enzymes are listed here. Now, these enzymes are coded for by genes like MOA. So ammonium monooxygenase is, for example, coded for by MOA, or these are hydroxylamine oxidoreductases, which are called as the HAO clusters, or these are called as nitrite reductases, so on and so forth. Now, the purpose of knowing these genes is that anytime when you are trying to follow the process of oxidation, the process of nitrification in a bioreactor, you can actually quantitate what group of microorganisms are present, what kind of biochemistries are going on, because you know what are the essential genes which are responsible for producing these enzymes. So through qPCR, through quantitative PCR, okay, you can raise primers against these genes, and then once you do you take a sample out, for example, extract the DNA, and then through this through MOA, you have primers which are very specific for the enzyme ammonium monooxygenase. If you do a qPCR of this sample, extract the RNA and find out how many RNA transcripts are present with respect to ammonium monooxygenase using these specific primers. You can also quantitate that how many of these microorganisms are actually occurring, because sometimes just measurement of nitrate, it is still at a very low level, which you cannot monitor by way of analytical techniques which are available. Okay, but if you know what kind of genes are carrying out these processes, which is indicative of the group of microorganisms which are involved, you know whether biochemically these reactions are occurring in the bioreactor or not. So is that point clear? Why is there emphasis on knowing what enzymes are responsible, what is the biochemistry, or what kind of genes are responsible? Because these are the genes you can raise primers against, and you can do a qPCR analysis, and you can figure out whether such kind of microorganisms, such kind of biochemical activities are occurring in the reactor or not. If there is a course correction required, if you need to make a fresh inoculum, or it is going, the bioreactor, is going in some other direction, you can still correct the course of the bioreactor. So is this clear to everybody?
Yes, ma'am.
All right, so this is a very, very important point, because when we do the real case studies, as you will see, we will again refer back to these particular genes that people have used the primers against these genes for doing a qPCR and finding out what is the population of, for example, Nitrosomonas, which is a very key, key microorganism which is responsible for this nitrification process, or nitrosococcus, or many of these microorganisms which have these particular enzymes, and they are part of the nitrification process. They can actually be quantitated by doing a qPCR analysis. So this is your first leg, which is called as nitrification. Okay, then the second part is called as the denitrification. Okay, by denitrification, what we mean is that nitrate is going to be converted, as you see in the last row here, it's converted into nitrogen. Okay, and all the reactions which you see which are occurring in between are reduction processes. That is, in the first step, the nitrate is going to be reduced into nitrite. The reaction is carried out by the enzyme nitrate reductase, and there are different groups of microorganisms here which are listed in the last column, and what genes are responsible which are coding for these activities are also listed here, so as to allow you in future to carry out, if you want to carry out a qPCR in the bioreactor, to know whether these groups of microorganisms are present or not, you can carry it out. Okay, so the first reaction in converting nitrate into nitrogen is conversion of nitrate into nitrite by the enzyme nitrate reductase. And since it is reduction, not only this process, but everything which is shown in gray color here, okay, all of these are reducing events. These are reduction reactions. In the next step, nitrite is converted into nitric oxide. This is the enzyme. Then the nitric oxide is converted into nitrous oxide. Again, it's a reducing event. And finally, N2O is converted into molecular nitrogen. Okay, so in the first process, for example, when I talked about the nitrification and the denitrification process, these are the reactions which are occurring. So this part is the nitrification, this part is the denitrification process. And what have you been able to achieve? You have been able to return nitrogen in the form of molecular nitrogen. So you have now added back that nitrogen into the environment. That's why we say it is nutrient recovery. Nitrogen has been recovered, otherwise it would have just been in the soil. Okay, and you cannot trap that molecular nitrogen, the nitrogen gas, into ammonium. Of course, it will be there. All right, ammonium will be present in the soil, the plants can take up, and so on and so forth. But then many of the plants, they also depend, we do depend on this reservoir, the nitrogen which is in the air, to be fixed by these microorganisms. And if nitrogen is not returned back into the environment, then obviously, after some time, this nitrogen fixation will just come to an end, because those are not there. Those groups of microorganisms are not there. So I hope you understand that why is it essential, therefore, to recover this nutrient. Okay, yes or no? So is this point clear to everybody?
Yes, ma'am.
So it is just, giving you an idea of some denitrifying species which have already been found in activated sludge. And mind you, in this process, you see so many genera which are there, which automatically, this is what we are learning, because this is constantly occurring in nature, not even in the wastewater treatment plants which we have set up. But if waste is left untreated in some other part, you will see that there are microbial activities which are constantly going on in nature, and so many genera are involved in it. Okay, so now, depending on the influent properties, okay, and the treatment configuration and the operating conditions, the heterotrophic denitrifying bacteria account for approximately 20 to 80% of the all bacteria which are dispersed in the activated sludge. Okay, so by influent property, what do we mean? That is, what is our ammonium load, what is our nitrate, what is our COD, what is our BOD, etc. What kind of treatment, do you want aerobic treatment? Do you first want anaerobic, whatever? And the operating conditions, that is the pH, the temperature, the external carbon source, etc. Okay, so they do make up, these denitrifying bacteria, they can make up as much as 20 to 80% of the bacteria which are found in the activated sludge. Okay, so is this point clear to everybody? So they make up a very large percentage of the bacterial population. However, they are still not large enough, because the conditions which operate under activated sludge process, which are highly aerobic, they do not allow these, the reducing activities which I showed you earlier. that for these steps to occur, up to nitrate is not a problem in the activated sludge process, but other steps, okay, which require a reducing environment, they cannot occur in the activated sludge process because you are dealing with activated with the aerobic tanks. Okay, and that is the reason why the population of these microorganisms is not large enough for you to be able to recover the nitrogen completely in the activated sludge process. So therefore, you need to either set up a reactor before or set up a reactor after that, where you can provide the desirable conditions for these groups of microorganisms to occur. Now, the second point, that most of these denitrifiers, which are in the activated sludge, are facultative aerobic bacteria. Okay, so by facultative, that means they can either grow under anaerobic or aerobic condition. Although they prefer molecular oxygen, they can use nitrogen oxides as electron acceptors, and therefore the absence of oxygen or the presence of an oxygen gradient within the activated sludge block is necessary for heterotrophic denitrification to occur. Okay, because all the four reactions which you saw in the previous slide, they must occur within with absence of molecular oxygen. They are all reducing activities. So therefore, for example, granule formation is considered to be very, very important in the wastewater treatment plant. Why? Because in the granule, in the flocks, there is actually an oxygen gradient. There is a lot of oxygen which the surface can make a contact with, but as you move inwards to the center of this flock or to the granule, the oxygen gradient decreases, and therefore there the denitrifying bacteria can occur. Okay, so all of these are very, very critical points which need to be kept in mind when you talk about recovery of nitrogen from the sludge. So, we use the term like heterotrophic nitrification. So now, why do we use the term heterotrophic? So here is the second leg of the reaction, where, because I will go back and very quickly remind you that ammonium to nitrate is an oxidation process, but this part that is going from nitrate to molecular nitrogen is something we said that it is a reduction process. So who is going to give you the electrons for this reduction? Okay, this is an organic or an inorganic carbon source which has to be added in the system, which can provide electrons, because that is how, for example, when you respire your hydrogen or the reduction process is carried out by availability of hydrogen from the carbohydrates. Okay, so in this process, the reduction, the electrons, for example, here, they need to be provided by an external electron donor. Okay, and that is why a carbon source is required. So if we look at this part of the reaction, converting the nitrate into molecular nitrogen, you see that a carbon has to be going in. So this carbon will be oxidized to carbon containing compound will be oxidized to carbon dioxide and water, and nitrate is going to be reduced now into nitrite. Similarly, when nitrite is reduced into nitric oxide, again carbon compound is oxidized to carbon dioxide and water. Okay, so we say, therefore, what we are trying to say, that in this part of the leg, okay, you do require external source of a carbon. All right, so is that part clear to everybody? Although there is a certain COD load in the wastewater, but that COD load is not enough to be able to support these reactions. That's why an external carbon source is required, because it is required at each step during the reduction process. So here you have like a summary of the whole thing. Carbon source gets oxidized to carbon dioxide, and you have nitrate, and these are all the different enzymes which participate in the process. Okay, nitrate reductase or nitric oxide reductase, nitrous oxide reductase, so on and so nitric oxide reductase, nitrous oxide reductase, so on and so forth. So in this process, for example, the electrons are collected from the carbon source, and these are now given to these for reduction, and your carbon source gets oxidized to carbon dioxide. Okay, so the basic requirements in this heterotrophic mode is that an external source of carbon has to be there. Okay, and so this can come from the COD, because the wastewater anyway they have a certain chemical, they have a certain COD component, and you can supply external carbon, so that the rate of, and also the rate of degradation of carbon, how rapidly under those conditions where you are operating the reactor, these carbon compounds can be degraded. Okay, and then the process nitrification also depends on the COD to nitrogen ratio, and what has been estimated is that you require 5 gram of biodegradable COD is needed per gram of nitrate which is going to be reduced. It depends on the dissolved oxygen in the system, it depends on pH and temperature.
Ma'am, so does it mean that a carbon COD available in the wastewater is less than is less than such that a COD and nitrogen ratio is less than 4, so that we have to supply external carbon source?
Absolutely, absolutely. So if you do the COD to nitrogen varies anywhere from maybe 1 or maybe to 2 maximum. And that is the reason why this denitrification cannot be achieved in the existing activated sludge process, because there is still a lot of nitrogen which just remains there unutilized, and the COD to nitrogen ratio is not enough. Okay,
Yes, ma'am.
so I mean, this is explained very briefly in this part here. Since organic carbon, which is naturally present in the wastewaters, is low, large amounts of externally added carbon is required. Number of carbon sources in under laboratory conditions, people have collected the wastewaters and tried to run it in the laboratory. They have used various organic sources such as glucose, such as lactate, such as ethanol, such as acetate. But they have also used an inorganic carbon source like methanol, for example. So externally added carbon source is required, and what is found is that if you use methanol as an external carbon source, there are a lot of advantages based on several studies which have been conducted. Number one, that about 2.47 gram of methanol is required per gram of nitrogen for complete denitrification, which is a much lower number compared to what you require through the organic carbon sources. So why methanol? Because methanol is number one, cheap, and it is a byproduct of the petroleum industry. When you carry out refining of crude oil into petrol, large quantities of methanol are available. So methanol is abundantly available as a byproduct of the petroleum industry. It is cheap compared to most other carbon sources. That is in terms of the chemical cost per mass of nitrogen which is removed. And number two, the methylotrophic organisms, which can actually use this methanol, because now when you supply methanol as a carbon source into the nitrification process, okay, then they are very slow growers, as a result of which it results in lower biomass yield, and that means also low sludge formation, and the related cost of residual sludge handling and disposal also goes low, because you are not generating too much of sludge anyway. Okay, so that logic you need to see as to why methanol is preferred over so many other. All of them can serve, glucose can be metabolized by the cells, lactate can be metabolized, ethanol can be metabolized, acetate. All of these are organic carbon sources. If you grow bacteria in the presence of acetate, it can certainly grow any one of them. But for methanol, only the methylotrophic bacteria can grow. So you want the slow growers, and you don't want too much of this carbon getting into the biomass. So low sludge is produced, and therefore associated cost of handling the sludge also grows low. And the third point says that methanol is utilized by facultative methylotrophic organisms. So therefore, these organisms are very slow growers. There is an acclimatization period varying from a week to several weeks is required in the wastewater treatment plant in order for the methylotrophic organisms to establish themselves. And similarly, an adaptation period may also be needed for other kinds of functional denitrifying groups. So that's the reason, before you want to do any startup activity in the wastewater treatment plant, we always talk about an acclimatization period. So if nitrification, denitrification is to be integrated along with the activated sludge treatment, normally you will see that, both these activities, that is the BOD removal by the activated sludge process and nitrification, denitrification is not carried out in the same vessel. These events are separated in space and in time. And a certain acclimatization period is required for these microorganisms to grow because they are slow growers, and as a result of which many of these activities can be carried out. So is that point clear to everybody as to what is the emphasis here?
Yes, ma'am.
So now, very quickly, I will tell you how. So you know a little bit about what is a microorganism, what kind of reactions are going on, and how these reactions are going to be dependent, the biochemistry of the reaction. So let us see now. This is, for example, from an actual process. Which is already integrated into the existing wastewater treatment. It is called as a modified Ludsack-Ettinger process. There are so many of them in the literature, but I thought some of the key ones I have just put down here, because otherwise we will have to spend many lectures covering this part. So what you see here, for example, is the influent. All right. So normally the influent, after the primary clarification, etc., it is directly fed into an aerobic vessel where activated sludge is treated aerobically. But what you see here, for example, all right, is an anoxic step which is preceding the activated sludge process. Okay. So by anoxic condition, what do we mean? How is anoxia different from aerobic and anaerobic condition? Anoxic means there is only absence of oxygen and other oxidizing agents. Okay. And aerobic means you do have molecular oxygen. And when you say anaerobic condition, anaerobic means there is complete absence of any external electron acceptor. There is no oxygen, there are no nitrates, there are no sulfates, etc. So there is no external electron acceptor under anaerobic condition. Nitrates are not present, and anoxia means lack of oxygen. Okay. And so therefore, other oxidizing agents such as nitrogen oxides, etc., can be present. It only means absence of oxygen. It doesn't mean absence of other electron acceptors. So in this particular, for example, step, as you see here, so this is called as a denitrification step. Okay. And denitrification, I will quickly take you back. Okay. So this is the denitrification step, where you don't require oxygen at all, where nitrates are converted into nitrogen. This part of the leg is called as denitrification, and this part is called as the nitrification. Production of nitrate is called as nitrification. Okay. So we say that now the influent is taken and it is first fed into an anoxic compartment, and here organic carbon is provided for denitrification. All right. And when complete denitrification is achieved, 75% of the influent nitrogen is already removed as nitrogen, and 20% of the influent nitrogen is incorporated into the sludge. So after this treatment, you can go now into the aerobic vessel. So the nitrogen has been taken care of under these conditions, and then your influent is now fed into the aerobic vessel where activated sludge process is applied, and through oxidative bacteria, your BOD and other things are completely brought down to the desired level. It goes into a secondary clarifier. Okay. And part of it goes here, and quite a bit of it is also recycled back. And if it is still rich in nitrate, then it can go back here and it can again come back into the anoxic part of the leg. So it's a two-stage process here. First the anoxic process and then the aerobic process. So through this clarifier, the activated sludge, for example, is returned back here. All right. So this part we call as denitrification, and this part we call as the nitrification process. Okay, so I hope it is very clear to you. So when this complete process is occurs, for example, the details are presented here, that 75% of the influent nitrogen is removed by emitting as nitrogen, and 20% of the influent nitrogen is incorporated into the sludge, and it is removed as waste sludge, and only approximately 5% of the influent nitrogen now remains in the effluent. Okay, so what you get in this effluent is just about 5% of the influent nitrogen which has entered into the wastewater. Okay, so I hope this you can see what we are trying to say here, and you're able to understand. So is this clear, please, to everybody? Okay. Now the second one, which has been applied, is called as a sequencing batch reactor. Okay, where the same steps are being carried out, but it is carried out in the same vessel. The vessels are not different. It is in the same vessel, but with respect to time, what is happening in that vessel is changing. So we say all these events which we are looking at in the same vessel are separated by time. So the first thing which you do is that there is a single vessel and the wastewater influent is coming in here. Okay, and then it is allowed to fill in, and this is an anoxic mix. So the same thing which you are doing here in a separate reactor is actually carried out in the same vessel for a certain amount of time. And then it is after these, let's say 2 hours of anoxia time is given, after that air is introduced into the same vessel. So now it becomes an aerobic mix. Activated sludge occurs, the waste sludge is removed, it can be recycled back. And then after this aerobic process has gone, which is on a time scale of 2 to 4 hours, for example, then in the same vessel, you allow everything to settle down. Okay, and the effluent is completely removed, and it could be idling because there is still a lot of sludge which is in there. Then the process could start again. Okay, so the sewage is introduced into a reaction tank, one batch at a time, and the wastewater treatment is achieved by a timed sequence of operations which occurs in the sequencing batch reactor tank. So you have filling, you have anoxic mixing, you have aeration, you have settling, decanting, so on and so forth. So is the difference between the two understood by everybody? And what you are trying to achieve under anoxic condition, what are you achieving under aerobic conditions here?
Yes, ma'am.
Yes, ma'am.
Okay, so very quickly, in that list, I put down 3 different processes. One was your nitrification and denitrification. Second was the anamox. Third was the demo. So in the anamox process, which is supposed to be taking up quite fast, especially in the developing countries, is the anaerobic ammonium oxidation. Okay, first of all, it's a single reaction where what you are actually going, the final reaction is shown here. Okay, that is the ammonium is oxidized under anaerobic condition where nitrite comes in as an electron acceptor. Because remember, when you carry this reaction out under anaerobic condition, molecular oxygen is not available for oxidation of ammonia. Okay, but instead nitrite. Okay, so you have a case where there is something else which is coming as an electron acceptor. So ammonium is oxidized into nitrate, okay, and molecular nitrogen. So in a single step, what you are looking at is that from ammonia, you can derive, for example, at least if you combine both of these stoichiometrically, there is nitrate is certainly produced because of oxidation reactions. But at the same time, you have 1.02 divided by 1.26. If you look at the nitrogen biomass is of course very, very small. Okay, so quite a bit of this, almost 80% of this ammonium is directly converted into molecular nitrogen. So it's a single step conversion which you are achieving under anaerobic condition. Okay, and what has been found is that of course the process is very, very sensitive to nitrite And if you have more than 50 to 150 milligram nitrogen per liter is inhibitory to the process, and it is estimated that globally this process could be responsible for 30 to 50% of the nitrogen recycling which is occurring in the oceans. There are specific group of bacteria which have been identified which carry out these anaerobic ammonium oxidation. Okay, and what is the advantage in this process? That first of all, it reduces the oxygen demand. In the earlier process, you were doing complete ammonium is converted into nitrate, which was an aerobic process. So the requirement of oxygen is very, very huge. But in this case, we will see that, the requirement of oxygen is very, very low, because in the initial process, okay, very quickly pay attention to it, because in the wastewater stream you do have ammonium, you could have nitrate coming in, okay, but then what you require in this particular reaction, what the microorganisms require are not nitrate, they require nitrites, okay, and for that first oxygen has to be supplied in the reactor or somewhere else whereby ammonium is partially oxidized into nitrite. Okay, and that's why we say partial nitrification, because if it is complete nitrification, then ammonia would combine with oxygen to give rise to nitrate. But in the final reaction, what you require for anaerobic oxidation is the nitrite. So in this process, therefore, partly ammonia must be converted into nitrite. This nitrite can serve as an electron acceptor for ammonium ion to give rise to molecular nitrogen, and the complete reaction is actually given here. Okay, so let's see what is the advantage of anaerobic ammonium oxidation. Firstly, this will reduce the oxygen demand because you are only carrying out partial nitrification. Number one, organic carbon which you require in the denitrification process. Remember, your process in the first process, nitrification, denitrification, is dependent on addition of external carbon. Here, that organic carbon or inorganic carbon is not required at all. So therefore, the biomass yield is very, very low. Consequently, very little sludge is produced. And this is another factor that contributes to substantially lower operating cost of this particular method. Okay, so these are, this is not a technology which you have developed. As we see here that globally, people have identified these groups of microorganisms which are carrying out this ammonium oxidation under anaerobic condition. All we are trying to do is to understand about the physiology of these microorganisms, collect them, make a consortium, get them acclimatized, and put it into a process whereby we can carry out in our reactors. So is this point clear to everybody?
Yes, ma'am.
Yes, ma'am.
Okay, so I hope you have understood too many things happening at the same time, the nitrification, denitrification, the first leg being aerobic, the second leg being anaerobic, then the second process is the anaerobic ammonium oxidation. What are the advantages, what you need to do, because eventually in the reaction you do require nitrite. Okay, so the nitrite needs to be generated by partial nitrification of ammonium. So some oxygen will have to be supplied to give rise to convert ammonium into nitrite, which comes in as an electron acceptor here. And then quite a bit of ammonium is directly converted into molecular nitrogen. So you are recovering nitrogen, you are putting this resource back into the environment.
Ma'am, your slide has gone.
Yeah, it's okay. I'm
Yes,
just
ma'am.
coming back
Right, ma'am.
again. Yes, I'm coming back again because I did notice that the slide has okay. So has it come back now?
Yes, ma'am.
Yes, ma'am.
Okay. I see. Uh, I don't know why okay, so let's move now to the next slide. So very quickly, what is the biochemistry of the process? And then we will look at, where exactly this AMOX reactor is operating and some very interesting figures which we have got. So if you look at the biochemistry, first of all, many of these microorganisms which carry out anaerobic ammonium oxidation, they look somewhat like this. A schematic diagram of this microorganism will look like this. And just like you saw vacuoles in the plant cell, these particular microorganisms contain what is called as anemoxosome. Okay, and there is a proper membrane here, just as a membrane separates the nucleus, for example, from the rest of the cell. In the similar manner, this anemoxosome also contains a proper membrane. And many of the enzymes which are in the process of this anaerobic ammonia oxidation are actually localized within this membrane. And this membrane is called as anemoxosome membrane. Here is the cytoplasm, and this is the outer layer of the cell. If it has a periplasm, then this is a cell wall, so on and so forth. So this is what the typical cell looks like. Okay. Now let us see what kind of enzymes and what are the kind of chemistries which are involved here. So quickly look at, this is your anemox membrane right here. This is the periplasm of the cell. So this part is certainly the interior of the cell, interior of the anemoxosome. Okay, so is this clear? This part is interior of the anemoxosome, and this part here is the anemox membrane. Here is the periplasm, and this is the outside of the cell right here. So from outside of the cell, molecules like ammonium, they are diffusible, they are readily diffusible. So they get into the periplasmic space. Okay, where, for example, the ammonium, what you see here, now the nitrites, the ammonium gets converted into nitrite. Okay, that step they have still not been able to figure out. But what they see is that the nitrite here in the first process gets, because even if ammonia needs to be reduced by nitrite, okay, the enzyme which converts the nitrate into nitrite. I hope that this is, yeah. So it is the same enzyme, nitrate reductase, which is a soluble enzyme coded for by the gene NER-S, which is shown here. Okay, and this nitrate reductase not only converts nitrate to nitrite, but it also converts the nitrite into nitric oxide. All right, so in the reaction where you have the nitrate getting converted into nitrite into nitric oxide, the second enzyme, what it does, that it combines with ammonium ions which are coming from outside of the cell into the anemoxosome. They combine with nitric oxide. The enzyme here is a membrane-bound hydrazine hydrolase, and they make hydrazine, because hydrazine has also been detected in these microorganisms, and hydrazine cannot be made by combining ammonium with nitrite. Hydrazine can only be made if nitric oxide combines with the ammonium ion. And this enzyme, if you look at its substrate specificity, we see that this can only combine with nitric oxide. So we believe, and I think experimentally it has been already proven, that the nitrate reductase carries out the conversion of nitrate into nitrite into nitric oxide. So then N2H2 hydrazine is released, then HDH, which is a hydrazine or hydroxylamine oxidoreductase, acts on hydrazine and converts it into molecular nitrogen. And that is how nitrogen is pumped out. All of these processes are linked to reduction or oxidation. For example, 1 electron is required for this reduction, 3 electrons are required right here, and finally an oxidation step. All of these are coupled to respiratory components, various cytochromes, ATPases, etc., coenzyme Q, which are located in the membrane of the anemoxosome. Okay, so is this step clear? That of course, collectively in the last slide we do pick up that ammonium is converted into nitrite, etc. All right, but you also see that from nitrate, nitrites are converted, and this doesn't directly combine here, but it gets converted into nitric oxide by some group of enzymes. And the stoichiometry is not known of that reaction, but it is known that there are enzymes which can combine nitric oxide directly with the ammonium ions to give rise to hydrazine. And that is what eventually what you see, nitrogen is coming out from the hydrolysis of hydrazine. Okay, so I hope this the biochemistry is clear, that what are the basic events which are occurring, and on a gross level, when you try to explain, when you try to understand the stoichiometry, only in terms of these constituents you are able to put it down in this form.
Yes, ma'am. So, ma'am, for doing this process, Anamox reaction, also we need to provide microorganisms which convert ammonia into nitrite.
Absolutely, absolutely. And just stay with that point, because now, for example, here is a full-scale Anamox reactor, okay, which is operating
Can
in
you go to the last slide?
previous one.
Yes, yes, ma'am. In this one, so
Mhm.
ma'am, in this anamoxosome, there is ATP synthase. So, like we know that, like in bacteria, ATP synthase is found in this membrane, or in mitochondria it is found, or like in
That's yeah, but
chloroplast
remember,
it is found. But in what purpose it is here in this anamoxosome? Also,
because
it should be
see,
like
yeah,
double membrane, then it works like ATP synthase.
certainly. So you see, ATP synthesis or biological respiration in these bacteria does not occur only through the anamoxosome. In the cell membrane, this is your cytoplasmic membrane. And remember that these are prokaryotic organisms. They don't have well-defined organelles like mitochondria. Mitochondria is only present in eukaryote, but but these groups of microorganisms, they don't have mitochondria. Okay, so when it comes to ATP generation, it is the anamoxosome membrane which you see here, and it is also the cytoplasmic membrane, which the different cytochromes and other things are localized, and through which ATP formation occurs. So this is not the only way the ATP is produced. Even in the site, there must be, some other carbon sources which are coming in, which it is able to metabolize, and for which it requires those cytochromes which are localized in the cytoplasmic membrane. And even in this anamoxosome membrane also, all right, there are these ATPases which are localized, which are able to provide energy to the cell.
Ma'am, also, like I did not clearly understood what is the role of this anamoxosome in ammonia conversion.
Because these special group of microorganisms are the only ones. What you are trying to say are that the reactions of the anamoxosome, okay, for example, are localized within this kind of a structure. Just as, like, in the plants you saw that when it comes to metal sequestration, all the metals are eventually stored, or they are carried out only in a specific part of the cell. In a similar manner, the reactions which are associated with anaerobic ammonia oxidation are localized, or they are coupled, for example, to only anamoxosome. In the rest of the cell, these reactions are not carried out. First of all, ammonium is toxic. Too much of ammonium is toxic anyway. So this is a way to localize, for example, ammonium or to nitrate ion to a very restricted part of the cell. Because the compartmentation, okay, in a prokaryotic cell, you don't have too many compartments. Everything is very, very fluid. But eukaryotes are more evolved. There is compartmentation. And by compartmentation, you try to segregate a group of reactions together so that they are not limited. Reactions are not going to occur only by way of diffusion.
Yes, ma'am. But like in the figure left, we are seeing that these reactions are happening with enzymes, with the help of enzymes in the periplasm, right? At
Right,
the
right.
periplasm.
Exactly, exactly. So this periplasm, for example, if you go back, is for example, in this part, when it is very, very close to the periplasm, because remember that this is, of course, the cell wall. Okay, this is the cell membrane, what you see here. the cytoplasmic membrane is very, very thick. So many of these reactions are going to, because this is just a schematic representation, and so this part is extremely close to the periplasm here. So the idea is that the ammonium or whatever, as soon as it is taken, so this is only one anemoxosome, but there are multiple anemoxosomes which are located right beneath the cell membrane. Okay, and their idea is that the ammonium is directly taken up by these enzymes, because remember, if you go back here, all right, so some enzymes are periplasmically located, some enzymes are membrane bound, for example, and this is anemoxosome lumen, which is the cavity, and riboplasm is actually what is outside of that. This is the cavity of the anemoxosome, and the periplasmic space makes a lot of close contact with this. I think especially if you try to see that this is periplasm, this is the cavity of the anemoxosome. So these are very, very closely located to each other, so that as soon as ammonium is taken up, it goes directly through the periplasm, it undergoes a conversion, and all the other reactions which are occurring are spatially very close to each other, so that they don't enter into the rest of the cellular cytoplasm, because this is riboplasm. That means riboplasm is right here. And the reason they call it riboplasm is maybe because it is very rich in ribosomes, all that, compared to, that's why they don't call it cytoplasm, they call it riboplasm. It could be very, very rich in ribosomes.
Yes, ma'am, maybe.
Okay, so these are some of the reactions which are occurring. And now coming back to nitration, so here is the actual data which is obtained from a very full-scale anemox reactor, and the capacity of this is about 70,000 liter capacity. And what you find here is that this is a cross-section of the reactor which is operating. It's fully functional in Denmark, so the cubic capacity is quite high, like you see here, all right. And it has a lower compartment, this reactor, all right, which is called as a lower compartment. The capacity is about 40 cubic meter. And then you have the upper compartment here, which has a capacity of 30 cubic meter. It is separated into two different compartments, what is called as an upper compartment and a lower compartment. Okay, and lower compartment is the one into which the influent, like you see here, the influent is now fed directly. So this is called as some kind of, these are settling plates, so that some of the sludge, etc., which comes out of partial nitration is retained on these plates, and then the rest of the influent is actually fed into this bioreactor. Okay, I will take you one step before this. So the sludge that you have, for example, which contains ammonium nitrogen to the extent of 1200 milligram per liter, is passed through what is called as a partial nitration or Sharon reactor. Because remember what we said, that it needs to be partially oxidized before it can enter into the anemox reactor. Okay, so that is what is shown here, that the domestic sewage, which contains about 1200 milligram per liter of ammonium nitrogen, is first fed into a Sharon reactor where a partial nitration is carried out, so that quite a bit of ammonium is converted into nitrite. Both ammonium and nitrite now enter into this. So a little bit of sludge is generated because cells are being produced. So they are collected on this. These are plate settler, some transverse plate settlers. So the sludge is going to come down. Okay, and the clear influent liquidy part is now allowed to go into the lower compartment. Okay, mixing is achieved, for example. So first of all, you have the liquid which is rising up, which comes down. So mixing is achieved by this liquid which is coming down. All right, the gas is also coming down, and also by the influent which is coming at a certain speed. So in the lower compartment, mixing of the sludge is occurring. So the reaction is allowed to occur. Nitrogen is collected. So then, it is obviously going up. Then you have a gas and a liquid separator here. Okay, part of the gas can be recycled back for achieving the mixing. The gas, that is a nitrogen, is also collected. Okay, and then part of the liquid which comes here is again recycled back into the bioreactor. So this is the overall setting up, and this some data I'm going to quickly show you from this. All right, so I hope you are able to follow. I hope everybody is able to follow what is being said here.
Oh, yes, ma'am.
So now here are, for example, the data which are taken from this over a period of 750 days. Okay, so on the Y scale, what we are looking on the X axis, we are looking at the time of operation of the reactor just after it is inoculated. Okay, and what do we have on the Y axis here are NMOX cells. What is the cell concentration here? Okay, so now in the solid I what yeah here are all these symbols are explained here. So what you see in the square are the NMOX cell concentration as measured by qPCR. Okay, so through the sampling port, samples are removed from the bioreactor, and then you use NMOX specific primer. You do a qPCR. On the basis of that, you determine what is the cell concentration. So these are the number, that is how these numbers are obtained. So is this clear as to how you get this?
Yes, ma'am.
The squares are the cell number, and they are only determined by doing a qPCR. What are the primers which are used? These primers are very specific for one of the enzymes which carries out NMOX reaction. So qPCR is a quantitative technique. So based on the fluorescence, so the more the number of cells, the more fluorescence you are going to get in the qPCR reaction. So it can serve as an index of determining the cell number. And the straight lines which you see here, for example, the black one, or this black straight line, or this black straight line, these are the lines which are fitting the concentration during periods of exponential growth for estimation of growth rate. Okay, so it is like, you are going to do, for example, this is the logarithmic phase of growth which you are looking at here, and this slope can be used to determine the specific growth rate. And once you have a specific growth rate, you can determine the doubling times. So the doubling times which have been estimated are 11 days for these microorganisms. So they are extremely slow growers. And what you are looking at here, for example, in the gray line is a 30-day moving average of the nitrogen conversion. So you are feeding so much of nitrogen into the bioreactor, but if you look at the conversion, these are the units, you are able to convert very little nitrogen, only 0.5 kilogram nitrogen per cubic meter per day. So what the figure is trying to tell you, that the growth, for example, of these microorganisms is very slow. During the first 750 days of operation, the nitrogen conversions are extremely low, and gradually, initially the doubling rates are, initially the cells will grow, then they will die out, but then later on they will re-establish, they will get acclimatized, and after that you see, of course, the slope here is less than here, but then now there is no lag per se, that the cells are already acclimatized. The number of cells which you are getting in this time period is much more compared to what you were getting during this period. So here is an example of acclimatization. So I hope that point is clear.
Yes, ma'am. Can you please explain again what is acclimatization? I could not understand.
Acclimatization is that, it is like getting adapted, getting used to something. So so it only means, the word comes from, the climate, because you can say that the cells are experiencing a very different type of climate inside there. Okay, normally in nature, they don't encounter, because these are NMOX bacteria which are generally found in the oceanic water bodies, and they are not used to seeing so much of nitrogen sludge, heavy sludge. So they are taking time to getting used to this. So during initial phases, for example, the growth is very slow and they also die out. However, some cells remain dormant, but then gradually they get used to that climate. Then they get started to grow again, And eventually, you see, the cell number which you are going to achieve after 750 days is much more compared to what you were doing at this stage. So this is only indicative of the fact that the inoculum which you have put in here is now acclimatized.
Yes, ma'am.
All right. So now nitrogen conversion in the NMOX reactor, for example. So here is over. So it was followed for a very, very long time. Like you see here, 1000 days, 1200 days. This is 1400 days. So 1400 days converted into years. So the progress of this reaction has been followed nearly for about 4 years, because these are very, very slow. But nevertheless, there is so much of advantage with them, like low sludge, low operating cost, no requirement of external oxygen, very low requirement of external oxygen, no carbon to be added in the reaction as an external source as you would require during the denitrification reaction. That it is now considered as a very, very successful thing to be to be carried out, to be integrated into the wastewater treatment plants. So in this particular one, what we are looking at is the conversion of total nitrogen, which is shown as a solid line here. Okay, so the solid line goes right up to here, which is approximately, the conversion is 640, about maybe like 750 kilogram of nitrogen per cubic meter per day. Okay, and when you read the paper, you will see that this particular plant was designed to give you a conversion of about 750 kilogram of nitrogen per cubic day. So it was able to meet the design targets. What you see in the solid square are, for example, the volume of the granules. Okay, so as the reactor operates, there is granule formation, and during this process, when the reactor is successfully operating, you can also see a lot of granules have built up. What do we see in gray, for example, is the relative volume of the granules. So the increase in the volumetric nitrogen conversion was always related to the availability of the granule. So ammonium is shown as a black thick line. So this line, for example, is ammonium. So during this part, for example, you see ammonium is very successfully, it is present in the influent, and eventually, the ammonium, of course, the system has achieved highest productivity, and then the reactor is also operated in a slightly different mode from 1300 day onwards, and that is why you see that some amount of ammonium is appearing here. Okay, and nitrate, for example, there is no nitrate here, but later on the nitrate also goes low in this side. Okay, so in other words, in the anaerobic process, what you need to see is that quite a lot of this nitrogen, which is in the form of ammonium, gets converted into molecular nitrogen, and that is indicated by the conversion term. So 750 kilogram of nitrogen out of 1250 kilogram of nitrogen in the form of ammonia, which is pumped into the reactor, okay, is getting converted. Okay, and after some time, excess of ammonium is just left out, because the system was only handled to do this much of conversion, and of course it is also operated in a slightly different mode after about 1300 days. Okay, so I hope you get a sense of what is happening here. So what are the summary of the points which we have learned in this is that in the conventional treatment plant, if you have wastewater activated sludge, then if you want to carry out this removal of nitrogen, there are two steps which are required. First is nitrification and the other is denitrification. Okay, and first part is aerobically, then a suitable electron donor has to be given. So the process is highly energy consuming, associated with production of excess sludge. It also produces significant amount of greenhouse gas, such as carbon dioxide. Why? Because externally you are adding a carbon source. Even when methane is added, methane is also oxidized into carbon dioxide and water. Okay, so you are generating carbon dioxide also as a byproduct. In the NMOX process, it is a single step. However, it is preceded by nitration. because you have to convert some amount of nitrate has to be converted into nitrite. Okay, and the process doesn't require aeration, other electron donors. However, still, as we said, oxygen is required, but the demand is greatly reduced. Okay, and of course, it has been shown that efficient application of the NMOX process in the wastewater treatment can result in the cost reduction by about 60%, as well as lower CO2 emissions. Okay, so is this clear to everybody? Because it's an extremely important point.
Yes, ma'am.
What about other students? Those of you who are logged in, do you understand what is being said here? Vivek Sehra, you are logged in. Are you listening to the lecture? Rajat? And there is no way that you can cramp up and learn everything just by reading yourself. So people have just logged in. Maybe I think apart from Ratnesh, Harshit, are you there? Kumar Kaushik?
Yes, ma'am.
So are there any questions on this, whatever we have discussed so far? Okay, if not, then we move to the next slide. And that was the third one. And I'm not going to spend too much of time on this. You just need to understand it only conceptually, that the third process of this nitrogen recovery was called as the demo process, which is the this is called as denitrifying anaerobic methane oxidation process. Okay, the summary of this process is written here. Okay, now in the NMOX process, what did you see? In the NMOX process, you saw that ammonium combines with nitrite in order to give rise to nitrate and molecular nitrogen. Okay, and that is the reason why
Ma'am,
this,
your
yeah.
slide is not changing.
Oh, it's not coming in. Okay, okay, let me see. Let me go back and then maybe. Is it clear now?
Yes, ma'am. Yes.
Okay. So let me go into the full slide mode. So in this anaerobic, methane oxidation process, what we are looking at here in the gray part of the leg, you will recognize this as what are the reactions which are occurring in the NMOX, which I just discussed with you. That is, in NMOX process, we have ammonium, which combines with nitrite in order to release nitrate and nitrogen. Of course, some part of our nitrate, we are converting it into nitrite. Okay, now the only difference here is that instead of adding an organic carbon like you saw in the other processes, okay, what we are doing is that we are going to use methane now, oxidize methane into carbon dioxide, and the processes which are conversion of nitrite into nitrogen is coupled to oxidation of methane to carbon dioxide. Okay, so that is the reason. So many of these events, we are not linking NMOX to DAMO. All we are saying is that some of the reactions which you have seen occurring earlier in the NMOX process, okay, that is, or in the denitrification process, whereby nitrate is converted into nitrite, into nitrogen, etc., or where many of these steps can be converted anaerobically, is something which can now be coupled to oxidation of methane. So methane will come as an external electron acceptor, and it will be able to convert nitrite into molecular nitrogen. So it is kind of a combination of demo and NMOX process here. So you require bacteria, okay, which will convert ammonia into nitrite or nitrogen. But then what provides for this reductive power are not the anammox bacteria themselves, but these are the methane oxidizing bacteria, which are going to provide these external electron donors. So the overall reaction when you see here is that you have methane plus you have now nitrate, which is generated, as a consequence of some of the ammonia is converted into nitrate also during the anammox process. So that nitrate can come in, they can be taken up by ammonia oxidizer, they can be taken up by methane oxidizers, and nitrogen formation can occur here. Okay, so these are methane oxidizers, except that they don't use molecular oxygen, but they use nitrate as an electron acceptor. And this is how much of energy that they are going to generate. And many of these microorganisms which can carry out methane oxidation under anaerobic condition, they can also take sulfates. They can also take metal oxides. Of course, in those cases, nitrogen is not generated, but then nevertheless, anaerobically methane can be oxidized. So I hope this point is clear to everybody.
Yes, ma'am. It is clear.
Now, the last part is here in this lecture is phosphate recovery. Why is it important? Because, phosphorus is also very important. There is a limited stock of phosphorus in the Earth's crust in the form of soluble phosphate. So if these phosphates are collected only as sludge, completely in the activated sludge plant, then there is no way that people are trying to understand that there is a limited stock of phosphate which is available in the Earth's crust. And after some time, the phosphates are not going to be available. So phosphate recovery is extremely important. It can be done either independently or along with, so many people have combined in a wastewater treatment plant additional reactors which can recover nitrogen and phosphorus simultaneously from the wastewater. So here is a picture, very quick picture, which allows you to understand that the source of it can be either organic, like is shown. So has the internet, can you listen to me?
Yes, ma'am, now we can.
We have lost the slides. Okay, but we have lost the slides, I think.
No. Slide is visible.
Slide is visible. Okay. So I'll just go back. Okay. So now I very quickly want to, before I tell you something about these bacteria which are able to recover these phosphates, is that first of all, phosphates are coming from both organic sources, biological sources, or they are coming from inorganic sources. From inorganic sources, what happens is that in the sediments which are in our Earth's crust, due to tectonic uplift, there is movement of these plates, the phosphate rocks are available. So through mining activities, through weathering activities, etc., these phosphates are available in the soil. Okay, now the soil erosion can occur, it can enter into rivers, oceans, water bodies, and then it can get back into the sediment. This is the inorganic route of where all phosphate lies. In the biological route or in the organic world, okay, through agriculture, that is, phosphates are available there, they are taken up by the plants as a nutrient, plants are eaten up by humans, but then humans are going to test out phosphates, and it accumulates in the sewer system, or it comes in the organic waste which we are secreting. It can end up in the agricultural field, so on and so forth. Or many of the industries are also utilizing phosphates, etc. Okay, and it appears, in the through detergents, etc., phosphates are liberated out, so on and so forth. So it is only to apprise you where all the phosphate can come from, either through inorganic sources, which are rocks, or through biological activity, in which the phosphate is really complexed with the biological sources. Okay, like you have ATP, etc. in the cells where phosphorus is present. You have nucleic acids where purines, pyrimidines, etc. They also contain phosphates. Okay, sugars are there, ribose 5-phosphate, etc. So all of that is the biological phosphate or the organic phosphate. Okay, so uh, the previous slide is important, so maybe I should explain it later. So, all right, let me just spend 1 minute on it. And if you don't understand, then please stop me. That what has been proposed is that if you want to recover these phosphate through chemical routes, not through using microbial technology, but through chemical routes, then in a typical wastewater treatment plant, there are several places where phosphate removal can be attempted. Okay, that's what we are trying to show here. Now, the upper part here shows you, for example, what happens in the activated sludge plant. So there is mixing, for example, the primary sedimentation, there is mixing can be carried out, and eventually it goes into the aerated tanks, which is called as activated sludge tank. Okay, then you go for secondary sedimentation. Now A will indicate that the effluent which now comes out of the conventional wastewater treatment plant can be taken forward for effluent treatment. Okay, or it can also be done, for example, when you collect the sludge. Okay, then at that time there is another route where, for example, B means it is the second place where the chemical treatments of phosphorus removal can be applied. All right. Or for example, in the primary sedimentation tank only, there is a lot of primary sludge which is collected, which is called as the raw sludge. Okay. And then you have the raw sludge which is going to be treated, etc. It is stabilized. And then the dewatered sludge is taken. Much of the phosphate goes into the process water, or the process water can be taken, and then phosphorus recovery can be attempted here. So I want you to pay very, very close attention to what is being said. In a conventional wastewater treatment plant, where you are not talking about biological modes of recovery of phosphorus, there are three stages, there are three steps where phosphorus recovery can be attempted by chemical means, which is either at the stage of effluent or at the stage of recycling of the sludge or the process water which is generated. Okay, and what are these chemical methods? These chemical methods are shown on the right. That is, either calcium salts or magnesium salts, etc., can be added. The process water, which is rich in phosphorus, okay, now it is stirred with these salts, and phosphates are now precipitated out as calcium phosphate. Okay, and I think magnesium is also some magnesium aluminum phosphate, something like that, or struvite magnesium salts, aluminum salts can be added. Okay, and the salts, the phosphate is simply recovered, and the treated water which is now discarded, because then you can do so many other things for recovery of this phosphate from here. The treated water which is depleted in phosphorus can be thrown out, or acid, base, sludge, ash which is generated here, okay, can be thoroughly mixed. And at this stage, there is a release of phosphorus of metals which is in the sludge or whatever. All of these by acid or by base treatment, they are collected. It is precipitated or residues are collected, and the clear supernatant is taken, from where precipitation, crystallization, liquid extraction, etc. can be carried out for separation of phosphorus in the form of calcium phosphate, magnesium aluminum phosphate, etc. And then whatever is left behind is just goes out. Okay, so these are the chemical points of treatment. So is this clear? If we quickly look at this slide, are you able to understand what it is trying to say?
Yes, ma'am.
We can have a doubt session. Actually, in the next two sessions, we again have our PPTs lined up. So I'm just trying to rush through. But we can have an extra class and look at the slides and see what you understand, what you don't, and we will come back. So the things which I discussed about in the last slide, they are all inorganic routes. But when you talk about the biological routes, Okay, the group of microorganisms which are going to be discussed here are called as Acinetobacter. Okay, and these are the bacteria which are called as phosphate solubilizing bacteria. They are the ones which can be incorporated into, or they can be integrated along with the normal activated sludge treatment plant. Their job is to remove the phosphate. Okay, so I think let me just stop here because we are rushing very important concepts. So I'm going to stop on this slide, and then I have maybe just 3-4 more slides which we will cover in the next lecture before we begin our PPT sessions. Okay, so is this clear to everybody?
PPT
Yes, ma'am.
beyond 28th and 31st, right?
Excuse me, what did you say?
When will be the PPTs? Like when are PPTs scheduled?
28th and 31st.
Okay.
Yeah, not in the next class. Yeah, next class I have another topic, which is risk assessment. Yeah, good for reminding me. So yeah, we have another lecture. So there I'm going to discuss about a part of what we are learning today, and also risk assessment, which is a very important topic in environmental biotechnology. Then our presentations are scheduled. So we actually have a class on 24th now, isn't it? So we finished a part of this, and then 28th and 31st we come back for our presentation sessions. Is that clear to everybody?
Yes.
Yes, ma'am.
Okay, fine. See you on Thursday then.
Yes, ma'am. Thank you, ma'am.
Okay.